Affinity chromatography is a technique used within biochemistry labs to isolate a protein of interest. So this is a technique that is used to specifically isolate a certain protein that you are interested in. Now what happens is that we have this column, and now this column has a specific ligand that is able to bind whatever protein we are interested in. Now, for example, hexokinase is able to bind to ATP. So ATP is the ligand. Now let's say this column is filled with ATP molecules. And now we add a solution of protein and we want to get rid of the unnecessary protein and we want to isolate our hexokinase. When we add in this protein solution to our column, the unnecessary proteins will wash out because they are not able to bind to the ATP. Whereas the hexokinase, it's going to get left behind and it's going to bind to that ATP. Now, if we take a look at the zoomed in version, we see this polymer, and this polymer has multiple ligands. And let's say these ligands are ATP molecules, and these green molecules are our hexokinase, our molecule of interest. So the hexokinase will bind to the ligand and it will not elute from the column. Whereas all the unnecessary proteins or fragments, they will elute from the column. And now we are only left with hexokinase in the column. How do we get rid of hexokinase from the column and isolate it into one of our test tubes? What we're going to do is that we're going to add a solution of ligand. So in our example, our ligand is ATP. So we're going to add a solution of ATP into the column. And what's going to happen is that that free ATP that's entering the column, it's going to compete with the bound ATP. It's going to compete for the hexokinase. It's going to compete for that protein. And it's going to wash away the hexokinase, our protein of interest, and it'll elute from the column and we'll get it in our test tube. So what's happening step by step? So a protein is added to the column, and in our column we have some sort of a ligand that is able to bind our protein of interest. So once the protein is added, we add the protein, the ligand binds the protein. The ligand will bind the protein and the unwanted proteins will exit the column. Now, in order for us to isolate our protein of interest now, because we're left with the protein of interest in the tube, we add a solution of ligand. The ligand competes for the bound protein and it will wash away our protein and our protein of interest is now eluted. I'm going to go back one more time and discuss our example of which ATP was our ligand, protein mixture was added, hexokinase binds to ATP, all the excess proteins are eluted, ATP solution is added, ATP solution competes for the hexokinase, it washes away hexokinase, and now we have our solution of hexokinase, our protein of interest. And that is affinity chromatography. It's based around the idea that specific ligands will bind to specific proteins.